In this video, we will discuss about this high nociceptive neuron relation with M2 type macrophages. In the previous video, we have understood the graphical abstract which shows that they have found out the inter-organ interactions of these cancer cells to create this tumor suppressive microenvironment. So, for that, what they have found out that is the nociceptive neuron that plays a role towards the M2 type macrophages. So, they started their studies with analyzing the patients who are affected with this head and neck squamous cell carcinoma. And what they did is they first analyzed this multiple immunofluorescence of the patient's tissues and compare them with this numeric rating system of this preoperative pain burden. And they are able to find out a positive correlations that patients who have high nociceptive neuron, which they validate using the biomarkers, has the high pain burden. Thereafter, they checked whether this nociceptives is also related to this high M2 type macrophages, which is a cause for tumor suppressions. And they checked this thing using the bulk RNA-6. And thereafter, they also did the spatial analysis using the RNA-6 system and this CODEX for this uh, head and neck squamous cell carcinoma patients. So in the panel B shows this bulk RNA-seq data. So they categorize these patients for this high nociceptive neuron and high or abundant nociceptive neuron and poor nociceptive neuron. And poor nociceptive neuron is represented by P, and abundant nociceptive neuron are represented by A. For the adjacent normal tissues, we do not see too much expressions of these M2 type macrophages, whereas for the case of tumors, and that too in the case where abundant nociceptives are found, they are able to see these M2 type macrophages are high. And these things later on they have validated using the multiple immunofluorescence image, where using the well-established biomarkers, where they validate that M2 type macrophages using the marker CD206 are expressed heavily in the case where this nociceptive neuron is evidently high and the pain is also high in the patients, compared to the case where nociceptive neuron is poor and pain is also low. And these things they have quantified on the right hand side of the panel B, where we do not see significant changes for M1 like tumor associated macrophages, but they see changes for this M2 like tumor associated macrophages, which is significantly higher for the case where nociceptive neurons are abundant and pain is also high. Afterwards, what I did is they checked it using the other biomarkers of this CGRP, which is also plays a role towards tumor suppressions, and this TRPV also, and they are able to see that the amounts of CGRP, TRPV, and other biomarkers that are associated with immunosuppressions are significantly higher in the case of this nociceptive neuron abundant region. And they have individually also measured some of the immune cells, such as CD45 immune cells, which are high, and the CD8 CD68 plus tumor associated macrophages are also high, and because of that the immune cells, cytotoxic immune cells, that is CD8 plus T cells, are also high. And these things they have checked for other T cells also, that is CD4 plus T cells, CD20 plus T cells, CD56 plus T cells. And what they are able to find out that in the case of this nociceptive neuron abundant, the formations the tumor associated macrophages are high that cause the tumor suppressive environment, and because of that the activated T cells seems to be gone down. And the quantifications of this panel D images We will discuss that thing in our next videos, where we will see how this immune suppression plays a role towards these different types of immune cells, how they what kind of immune cells that they inactivate, and what are the kind of immune cells that they do not affect at this point of time. We will discuss that thing in our subsequent videos, where we will see the statistical analysis of this panel D. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.